So I will talk about the title today. I will just jump right into it. Let me see if I can change slides. Yes, so I'm part of the nanoPASS project. It's a project that looks at new nano safety testing technology. It's anchored in the AOP framework, and we look at in vitro methods for key events. There are a lot of other people in this project, so I will only be talking about my work, but there is also partners working on advanced imaging of how these key events can be visualized in silico tools, and looking at in vivo data to calibrate the in vitro methods. We have 4 objectives, and my work fits into 2 of these: development of in vitro models relevant for prediction of in vivo adverse outcomes, which I will come back to, and calibrating and validating the in vitro outcomes to in vivo data. So here we look at in vivo data from historical data, but we also have case studies where we collect particles from cement, nanoplastics, electronic waste materials, and advanced materials for catalysis and medical applications. And these particles will be collected at different stages of the life cycle. So the method that I'm working on, we have to look at the respiratory parts of the lungs and going all the way down to the alveoli. They have a diameter radius of 0.2 millimeters, and this is constantly changing since we are breathing. And what I'm working on is actually a thin layer of liquid called lung surfactant that covers the alveoli. And the lung surfactant is produced by the type 2 alveolar cells and released into this liquid lining of the alveoli. As I already said, the alveoli and the surface area of the lungs is constantly changing since we are breathing. What is important here is that we have an air-liquid interface, which is covered by phospholipids that is depicted here in a blue band. So as you are breathing out, the surface area of your lungs becomes smaller, and this pushes the phospholipids into the liquid interface. We have 2 essential surfactant proteins, the SPB and the SPC protein, that functions by keeping the phospholipids close to the air-liquid interface. And they do this because once you are breathing in again, the surface area of the lungs becomes larger, and it's important that the phospholipids cover the air-liquid interface quickly. In the lab, it looks like this. We have a pedestal where we have the lung surfactant sitting on top. So this is an in vitro method that doesn't have any cells. It only has this physiological liquid. And the drop of lung surfactant is, the pedestal where the drop of lung surfactant is sitting on is hollow, and it's connected to a syringe that is filled with a buffer solution. And the syringe is controlled by a motor where you can control it by a computer program. So we introduce and remove liquid from the lung surfactant drop in the frequency and in the compression rate to simulate what is happening in the lungs. So it looks like this. Simulating a breath out, the droplet becomes smaller. And as you can see here, the drop becomes also more flat. This is because when you compress the lung surfactant, the surface tension is lowered. So if you had a drop of water, this would not happen. This is what is the effect of the phospholipids and the proteins. So you simulate that you're breathing in, the droplet becomes larger and the surface tension goes up. So this can be depicted in these kinds of graphs. We have a surface area change of about 20%, so in the physiological range of the lungs when you're breathing. As you can see, as you compress, the surface tension of the droplet goes down. And when you expand the droplet, the surface tension goes up. And this happens with every cycle of breath that you take. So the experiment looks like this. This is again a single cycle. We reduce the surface area of the drop and expand it, and the surface tension goes down. The drop is sitting in an exposure chamber, and then you can start to expose the drop to an aerosol where you want to test if the substance has any effect on the surface tension, the lung surfactant function. So this is the same drop exposed to a test substance, and what happens is you make the drop smaller and larger to the same extent as before, but the surface tension does not go down. So the minimum surface tension is what we are most focused on. So we have collected what happens if this would happen in the lungs in an AOP. This is AOP 302. So first of all, if you inhale something that can inhibit lung surfactant function by interacting either with the phospholipids or the proteins, if you don't have a low surface tension at the end of your out-breath, so when the alveoli are at the smallest, the alveoli can collapse. And this reduces the surface area you have in the lungs for exchanging gases between the air and the blood. It also reduces the tidal volume, so how much that you are breathing. But since you are still breathing, hopefully, this can open the alveoli that are collapsed. However, when this happens, it puts a lot of shear stress on the cells that are covering the alveoli, and then you can have a loss of capillary membrane integrity, which means that blood components can leak into the lungs, and the blood components will further inhibit the lung surfactant function. And all of this together leads to decreased lung function. So I will tell you a little bit about the background for why we look at the lung surfactant function in a case of impregnation products, the predictiveness of the in vitro methods that we have established so far, how we can combine it with exposure modeling and the planned work in nanoPASS. So first of all, impregnation products, they are used to make surfaces dirt and water repellent. They are a class of products that are used both by consumers and workers, and they are very often sprayed on surfaces. There are every year reports of human poisoning cases. After people have used these impregnation products, it becomes difficult to breathe, they start to cough, and they have a tightness in the chest. One of the things with this product is that they are often sold in pressurized cans, and they create aerosols that you can breathe into the lungs. And most of these cases, they recover with supportive care. So one case, this is a very typical what they look like. A person bought a new sofa and used 2 spray cans to spray the sofa. They had opened the windows and doors because it says on the cans that you need good ventilation. They used 30 minutes to spray, and they experienced during the spraying and straight after that they started coughing violently. They had a tightness in the chest and difficulty breathing, headache, nausea, and fever. So in this particular case, the person also had a bird cage in the living room where the sofa was. The person went to hospital to be treated, and when they came back, they also find that all the birds were dead. So the predictiveness of measuring lung surfactant function inhibition, we have looked at different classes of aerosols. So for impregnation products, as I've already told you, when you look at the lung surfactant function inhibition and compare it to breathing pattern changes in exposed mice, or when you compare it to human data where you have these accidental exposures, the sensitivity of only using the lung surfactant function measurements to predict is 100% and the specificity is 63%. We have done also measurements on pharmaceuticals for inhalation, both comparing to the breathing pattern changes in mice, but also human data for marketed drugs. And here the sensitivity and the specificity is 100% both. For single chemicals tested for GHS regulation, we looked at changes in respiration in rats. These were cage-side observations that were done either during exposure or within 2 hours of exposure. And the sensitivity was 81% and the specificity 100%. We have also challenged ourselves to use the in vitro assay to do risk assessment only using this assay. So we had 13 products for impregnation and 8 products for cleaning, because these are also often sold as sprays. We did exposure assessment in a chamber where we used the original packaging and estimated the alveolar deposited dose. And then we tested for lung surfactant inhibition. So this graph can probably be a little bit difficult to interpret, but on one side we have the impregnation products and on the other side we have the cleaning products. The dark gray dots are what we estimate that is deposited in the alveoli in the use scenarios. And the gray is the inhibiting dose that we found in vitro. So, and the line is a margin of safety. As you can see, for the impregnation products, almost all the dark gray dots are higher than the light gray dots, meaning that there is likely that there would be effect on lung surfactant function if this was used in this scenario. And for all the cleaning products, there was a large distance between when we saw an effect or we did not see any effect at all, indicated by arrows. So these likely do not have any effect on lung surfactant function. So the plan worked in nanoPASS is to test the particles from the industrial cases for lung surfactant function inhibition and compare it with the data that would be generated in the rest of the consortium and integrate this data into the weight of evidence for this AOP. Some of the challenges with this method is that we can estimate the dose, but it can be challenging with the different kind of particles or aerosols that we use, and it does not always compare with the in vivo estimated doses. Also, we only test one toxicological target in the lungs, so we just look at lung surfactant function, and it needs to be combined with other in vitro methods. So for example, some of the methods that Tommaso talked about earlier this morning. And with that, I hope I kept my time, and there is also time for some questions, and I would like to thank you all for listening.
Absolutely perfect with the timekeeping, and indeed there's some time for questions. So if anybody has any questions, then please feel free to sort of raise your hand now.
Otherwise, I will also be part of the panel discussion later.
Indeed. So as there doesn't appear to be any immediate questions, I suggest we move straight on to the next presentation. So coming up next, we have Alberto Katsumiti from GAIKER, who's going to be talking about the third project in the collaboration that's putting on this whole event, which is iCare. And he's going to be talking about that project's approach towards the harmonization of human and ecotox models for neurotoxicity. Alberto, straight over to you to begin. Thank you.
Thank you very much, Sean. Can you hear me?
Indeed, yes, and we can see your slides as well.
Very good. So put in presentation mode. So it's going quite slowly. Yeah, very good. So well, thank you very much for the opportunity to present here the iCare's approach towards the harmonization of human and ecotox models for neurotoxicity. So as you can see, just in the title of this presentation, so we are a little bit different compared to the other 4 projects of the same call. So this project is very focused on the neurotoxicity effects, especially the ones related with exposure to nanomaterials or advanced materials. So just to start, I'm showing these slides just to show the composition of the consortium. So as you can see here, we are 11 partners all around Europe. And we also have Australia in the consortium. So as you can see here in this image, Australia is in the north of Europe. So it's a funny map, but well, anyhow. So in the bottom part of the slides, you can see the main objectives of the project. So the main 3 objectives of this project is the development of imaging technologies or imaging methods to quantify physical chemical properties of advanced materials in complex matrices. And we also focusing on the development of new high throughput in multidimensional assays. That is, this assays is applied to the in vitro methods that we're developing, so which is the third part of this development set. So in this last part, basically, the objective is to develop new in vitro and also bridging models for nanotoxicity. So in this slide here, so you can see basically, this presentation is very focused on the in vitro and bridging models that we are focused on. So as you can see here, so we have 3 layers of development. So we have the human part, the bridging models, which is basically the interface of humans and also environmental, and then the ecotox models as well. So in the human part, so we have 2 main developments. So basically the first one focused on the development of in vitro human-based blood-brain barrier model for permeability studies. And we also have an in vitro human-based model to assess neurotoxicity. Bridging models, we're focusing in 2 species. So basically C. elegans and planaria. In ecotox, the ecotox part is very focused on the development of in vitro models based on fish cell lines. So we are talking about monotypic culture and also co-culture systems using, for example, immune cells. So going through these slides, especially like in the part of the human health based, so in this first slide, I'm showing like some, the initial strategy that we are applying. So we are in the very initial phase of this development. So basically, in this first part, we are basically exploring 2 types of models. So static models for the BBB and also a model based on microfluidic that you see like in the next slide. So in this first part of the development, we are testing, basically we're testing different cell lines to apply to use in these models. So in this first set, we are exploring the possibility of using just 2 lines. So for example, endothelial cell lines and also astrocytes. And then in the second part, we are testing 3 types of cells in the same system. So endothelial cell lines, pericytes and astrocytes. So we are testing different commercial cell lines, human-based, as I mentioned, and we are seeing different performances of the cells. So in the graphs that you can see in the right-hand side, so basically we have data on the tear values and also lucifer yellow to see barrier permeability, for example. So then you can see these are just codes of these different models, but we can see that some of the models, they have a better performance in terms of barrier integrity and higher values for tear measurements. And the same is seen, for example, in the lucifer yellow assay. So you see that some of the models, they show like better results than the others. So the idea is to really test the combination of different cell lines, human-based cell lines here, to then select the best ones to use in this static model. So as I mentioned before, so in the second part of this development of the BBB model, so we want to take the same cell lines, so endothelial cell lines, pericytes, and astrocytes, in case we use 3 cell lines, of course, in the first part in the static system. So we will apply these cell models in this microfluidic system. So this microfluidic system is a new development, as you can see here in the images. So these are a little bit different of the typical microfluidic chips that we are more accustomed to see, for example, in the microfluidic experiments. In these systems, for example, we're reproducing the veins, for example, and then we apply, for example, endothelial cells and astrocytes, for example, in the systems. And in the images below, you can see, for example, marked in the fluorescence, a red color, the endothelial cell lines. And in green color, we can see the astrocytes in the system. So you can see that the cells, they settle, they attach to the system, and they work quite well in this tubular system for the microfluidic system. So the next parts will be to use, for example, and to test the adhesion, for example, of the pericytes in the system as well. But so far, we have good results using these systems. So in the third part of the human health development, so we want to explore the possibility of using the same cell lines or similar cell lines, for example, used in the previous models, but applying here just as a neurotoxicity model in vitro. So the idea here is to test the same astrocytes, for example, in the system, but then apply also immune cells, so microglia, and also neurons in the system. So the idea is to test the possibility to have 3D systems, so based on inserts, so separating, for example, the different cell lines, but also to use a submerged system combining the 3 cell lines in the same model. So with this system, what we need to do initially, for example, is to characterize the responses. So we want to focus especially, for example, in inflammation. So the idea is to use, for example, Luminex to characterize a panel, for example, of immune responses in the system, but also look at production of glutamate and amyloid beta, as well as some potential specific responses in the system. Good, so moving towards to the bridging models. So in this first slide, you can see that the first bridging model that we're exploring here is the C. elegans. So C. elegans is a very interesting organism because have a very short lifespan, so of more or less 2.5 weeks. So as you can see, the majority of the human genes are conserved in this species, are present in this species, including disease genes. So this is a very interesting species because it serves as a bridging model between in vitro and also in vivo, but also because it's a bridging model between environmental hazard and also human health hazard assessments. 2 main things, 2 main directions, for example, with these bridging models have been explored. So for example, the possibility of the establishment of Alzheimer's disease strain. This is under development right now, and also the establishment of transgenic neurological strains to visualize the neuron degeneration. So as you can see here, for example, the 2 images below, we can clearly see, for example, that this animal, which is like a transparent animal, can express, for example, a normal condition. So we can see this very clear fluorescent signal. And then once we have the exposure to some chemicals that can induce, for example, neurological degeneration, for example, we can see a decrease in this fluorescent signal. Well, the second species, the second bridging model that we're exploring here are basically planarians. So these animals, they are being validated as an alternative in nanomedicine. We know that, for example, planaries have been used, especially, for example, in tissue regeneration experiments. So it's been widely used, for example, for this kind of research. They are also considered the earliest extant example of evolution of mammalian-like brain. So they are also widely used to evaluate the toxicity of metals and also organic contaminants. And again, similar to the model before, as you can see, that's these models, they serve as a bridging model between in vivo and in vitro, and also between environmental hazards and also human health hazard assessment. So 3 main points will be explored here using these animals. So basically, neurobehavior evaluation, so basically the movements of the animals. So it's a clear response that we can see, for example, in the exposure to chemicals. Also enzymatic activities, as you can see here, for example, acetylcholinesterase, ATPase, in MAO-A, and also effects on the effects related with cytotoxicity and also genotoxicity, based, for example, in the COMET assay. So the ecotox approach, for this ecotox approach, so the idea is to use in vitro fish cell lines for nanotoxicity assessments. So the idea is similarly to the human health. So the idea is to initially, for example, test the usability, for example, of different cell lines. So we are exploring cell lines from different fish species. So initially, for example, we are testing the rainbow trout cell line, which is basically being developed, for example, in Canada, which is a collaboration that we have, but also local species, European local species that are being explored as well is in the system. So the idea is to test the possibility, for example, of having a simple monotypic culture of these neurons, for example, or also the combination of neurons with immune cells, with microglia cells as well. Possibly, for example, of course, the same fish species. We want to explore, for example, the sensitivity and the usability, for example, very classical and general parameters, cell parameters that we can measure, for example, in vitro. So, for example, cell viability, plasma membrane integrity, cellular uptake, and oxidative stress, but also more specific effects that can be related with exposure to this chemical. So effects related with inflammatory responses, antioxidant enzymes, lipid peroxidation, acetylcholinesterase, and etc. So here, again, we want to characterize the inflammatory responses using the Luminex to see the expression of many different cytokines and chemokines in these cells. So here we can see, for example, basically the group of positive controls in materials that we use as for these tests. So we separate 3 different groups of materials. So positive controls, benchmark nanomaterials, and also advanced materials. So as some possible positive controls that we use, for example, tritone X for the barrier integrity and also cell viability. LPS, TNF-alpha, and other cytokines, for example, for inflammation. This is just some examples of the positive controls, but of course, many chemicals are being considered here as positive controls to these tests. Benchmark materials that we are using in this project. So we have here, so for example, silica particles from the JSC, so NM203. We have also silver nanoparticles from the JSC as well. And also graphene materials, also coming from the JSC. All the benchmarks are being considered as well, as I mentioned before, but the list is quite long, so I prefer to just keep like short in this presentation. And also a third set of experiments will be performed using industrial materials. So basically, advanced materials based on graphene oxides, reduced graphene oxides, advanced materials made of the combination of graphene oxide and also silver nanoparticles, and so on. So I decided to order this in these 3 blocks because for developments of these in vitro methods, we want to first set up the models, test against the positive controls, and then a second set of tests use the benchmark materials and then confirm these responses using the advanced materials made of these materials listed here. So this is a very short and very general roadmap towards the harmonization in the project, but you can see here in a very general line. So we have a first initial set of developments, which is basically where we are right now. So we are in this first part of the development. So basically setting up the models and also the assays. Then we will have, of course, intra-laboratory experiments to test the validity of the methods and the reproducibility of the methods as well. Third activity, of course, is to work on the SOPs. So to prepare very detailed SOPs. In a second stage of this development, we will go to a process of verification of these SOPs. So these protocols and also the cell lines will be shared in the consortium with other partners in the same project. So we will test these different models. We will test these different assays as well in this inter-laboratory experiments. And then we also verify the robustness of the SOPs and the level of detail of these SOPs. And then, of course, if we need to make some adjustments, for example, in the protocols or in the in vitro systems and also assays, we'll do this in this phase. And then the idea is to move towards like a third layer and maybe the fourth layer of, so the third layer, so the validation. So maybe to submit, for example, these SOPs and these protocols, these models to the ACFAN and also NetVol for this interlaboratory experiments and also SOP validation. And then, of course, we still need to discuss because, as I mentioned before, we are in a very early stage of the development, but also to move forward, for example, submitting these protocols to the CEN and also as a possibility to ISO and OECD for the validation of these models and assays. So in the project, as I mentioned before, so we have also we are also working in developments of different assays. So we especially focus on high content assays and assays that we can measure in real time. So here in this next slides, I'm basically showing some of the approaches that we have, like in the for this. So as examples of the high content assays, for example, we are especially interested in inflammation, genotoxicity, and also epigenetics. So for inflammation, for example, as I mentioned before, so the main method that we want to apply is basically Luminex to characterize, for example, the expression of different cytokines and chemokines. We also have approaches dedicated to genotoxicity, so exploring different mechanisms of genotoxicity, related effects, and also epigenetics. Real-time methods, for example, so in these steps, for example, we want to use high-content assays based on multiplex sensors for multi-biomarker measurements. So this development is ongoing. So I know that, for example, we are able, for example, to measure these parameters in a bigger system. So we're talking about plates, microplates of 24 wells. The idea is to reduce the size of these sensors to be able to do these measurements, for example, in plates, in systems, in smaller systems. So for example, 96 well plates. So the idea is to have a system able to measure in real time different parameters on the same time. So basically speaking about biomarkers related with oxidative stress, inflammation, and also cell death. And also high content analysis, basically integrating different omic analysis. So the idea here is to explore the possibility to integrate transcriptomics, proteomics, and metabolomic analysis. So then integrating this in these big sets of data for identification of key aspects related with neurotoxicity pathways as an early response compared to this also in vitro assays we have developed before. As I think a last development that we have is a high throughput screening. Is a development for high throughput screening. So we are working on a device for real time measurements of TIA values. So, of course, I mean, everybody uses, I think it's pretty common to use TIA measurement, for example, in the different labs, but normally most of us, we use sensors to measure each time. So this development that we have is basically an equipment. There are some commercial versions of this equipment, of course, but we are working on this in this project. So an equipment and also in a software able to measure tear values in real time, and also to measure in 96 well plates. So in this way, we can not only to measure these in real time, but also and to use this in a smaller system. So in inserts, using inserts of for 96 well plates. So the main idea here with these assays, and also at some point with in vitro models, to try to miniaturize as much as we can the systems. Because we know that, for example, some of these models can be used, for example, in 24 well plates, and also sometimes in 96 well plates. But as much as we can, we want to move towards a more high throughput system. So we are talking about plates, for example, of 384 wells. So with all these developments, for example, of course, we can't, adapt, for example, some of the systems to these very, very small systems, because, for example, at the moment, we don't have like inserts for, tiny wells as 384 well plates. But as much as we can, we want to reduce, and we want to make the systems smaller and more high throughput. So for example, in the assays developments, so for example, the biochemical assays, so we're basically working on the validation of the sensing platforms, as I mentioned before. So the idea is to reduce as much as we can the systems, especially the assay systems for the biochemical assays related with the absorbance, fluorescence, and luminescence detection. And also the cell-based assays targeting, for example, reporter assays, phenotypic related assays. So, for example, as indicators of toxicity, and high content screening assays. The whole idea is to have options, is to have assays that can be high throughputs, affordable, and easy to use. So all these developments related with in vitro models, bridging models, and also the assays, they need to be easy to use, they need to be affordable, and they need to be high throughput. So all the developments are very focused at the end, and to have these three, ticks marked at the end of this developments. So this is all from my side. So I don't know if you have any questions.
Thank you for that, Alberto. Are there any immediate questions for Alberto's presentation? And Barbara, you have your hand up.
Thank you. This is a quick question. So pertaining to the slide about your library of positive controls, etc., are they sort of candidates that you want to sort of investigate as as proper positive controls across your assays? And as well, have you sort of, are these candidate nanomaterials, are these candidate advanced materials, or have you actually verified, yes, these are our performance standards, and that's what we're moving forward with?
Yeah, I think well, the main thing, for example, with the positive controls is to ensure that we have positive controls that we can have a very clear response. So it's very focused on the sensitivity of the models that we have. So we want to ensure that we have a selection of positive controls that actually produce a very clear response in the different assays. And then the benchmark materials is also related with sensitivity. We want to ensure that these models that we develop for neurotoxicity, they are not only sensitive to the chemicals, to these positive control chemicals, but also to nanomaterials. And then a way to confirm the sensitivity is to test similar materials, basically, for example, advanced materials made of the same or similar benchmark nanomaterials. So then to test this real life materials, for example, if our models can be sensitive not only to these benchmark materials, but also to real life materials or products. So then this way we can confirm, the validity, for example, these models.
Oh, no, definitely. No, I understand what you want to use those materials for, but I just wanted to know at what stage are you at with the materials that you listed on that slide? Are you at the stage where you can say, yes, we can use these nanomaterials and advanced materials that you've listed in the slide as performance standards? And yes, we can use these positive controls, or are you guys still in the process of determining whether you can use those materials as your benchmarks moving forward?
Yeah, so as I mentioned before, we are very in an initial phase. So at the moment, we're very focused on the cell models that we want to use. For example, in these systems, we are starting to test some benchmarks, but of course, some of them they show a better performance, others not the best performance. But I would consider that most of the activity right now is focused on the cell lines that we want to use in the systems. So basically, we imagine very simple parameters. So for example, in the barrier integrity, so the TA values in lucifer yellow, for example, for permeability of the models. In initial phase, basically just measuring in a normal condition. So basically, we're not exposing the models to anything and just measuring the basal levels of these values, and then also against some positive and negative controls. But it's a very, very early stage of the selection of materials.
Definitely. Thank you very much, and good evening from Australia.
Oh, thank you.
Okay, Alberto, thank you ever so much for a good presentation. Just to let all the previous speakers also know that after Elijah's presentation next, we'll move on to the roundtable being chaired by Blanca. So if people could just, if all the speakers could get ready for that, and in 20 minutes time, we'll start the roundtable. But at this moment in time, I'd like to welcome Elijah Petersen from the other side of the world, from Australia, from the US. So again, a completely different time zone. And he's going to be talking about strategies to increase the technical quality of new approach methodologies and nanotoxicology tests. So over to you, Elijah. Thank you.
Okay, you can hear me okay?
We can indeed, and also see your slides.
Awesome. So I'm a bit under the weather, so pardon me if I'm coughing a little bit during my presentation. But first off, I'd like to express my appreciation for this opportunity to share about some of the work that myself and many of my colleagues have been working on. I'll be, I guess, talking about two somewhat related, but also a little bit distinct topics. One is some of our work to have to improve the technical quality of new approach methods, and also some of the work that myself and colleagues have done over the years with nanoecotoxicity tests at OECD. So to take a step back, I'm highly involved with ICVAM, which is similar to ECVAM in the EU. One of the things that we've been working on is a validation report. You may notice that we've had a validation report, but it's from 1997, and quite a lot's happened in the last 26 years. So we've been working in the past 4 or 5 years to update this to reflect the current state of the knowledge.
Elijah, just to say, I think if you want to put your slides into presentation mode, it's just because we're still on your first slide.
Okay, I'm not sure why that's happening. It looks like it's in presentation mode on mine. I have slideshow, and if I go forward, it doesn't change. Is that right? If it doesn't
No,
change.
we're still seeing, we're still not in presentation mode. I think if you unshare your screen and then share it, then take it out of presentation mode and share it, and then put it into presentation mode, it should work.
Okay, let's give it a go. So I will do a new share, and then is it showing it now?
No.
It went up for public commenting back in August, so some of you may have had a chance to review it and send in some comments. A key part of this report is this figure that was kind of refined from Petersen et al. A paper that was published very recently, and there's many components to having a good NAM, including biological relevance, data integrity, and the information transparency. What I'm going to focus on is really here the technical characterization part of it. So during I guess it's been published, was it earlier last year, I think, earlier this year, a technical framework for trying to add quality to NAM. So this is not specific to any NAM. It's more like a general process that one could go through to try to add quality to hopefully any NAM. This was very much interagency effort within the U.S. government and also internationally with EMPA in Switzerland. And I'd really say that the whole is greater than the sum of the parts here. There was a lot of unique perspectives. We had some regulators involved and so forth that, everyone brought, unique and valuable perspectives. So what we tried to do is to, of course, everyone wants reproducible NAMs that are give similar results across time and among laboratories, but how to get there, that's really the key question. One of the things that we'd recommended was to apply basic quality tools similar to those in quality manufacturing to assess NAMs. And as I'll describe in the next slides, we also described a process for adding statistical confidence to these decisions. I won't have a chance to get into that today, though, given the time limitations. And it should be noted that it's not like an infinite number of controls is the best way to go. There could be decreased throughput or higher cost for more controls. So it's important to capture the things that really matter, but then you could also, there's trade-offs. So if you have more than is needed, I guess. And that's part of where the consensus discussion process comes from. So this is a key figure from the paper, where in the beginning you have the world of NAMs, where there's, I don't know, 100,000, some huge number of NAMs throughout the published literature. And so one of the key parts is figuring out which ones we actually want to spend the effort to improve the quality of. So some of the key questions that we'd consider was, is there biological relevance, such as linked to an AOP? Could it fulfill a regulatory or non-regulatory testing needs, such as for a business trying to make a decision before to whether to go ahead for the product or not. And this is mostly, I should qualify that most of my discussion here is for laboratory-based NAMs. For computational NAMs, some of the considerations will be similar, but there's also some unique nuances to that differ from what I'm describing here. And third, is there a capacity to improve the quality sufficiently? It could be that if you spent $1 million or €1 million or what have you, that if there's some instrument that's very hard to calibrate, or it's only one of a kind and you need to show transferability, it may be that you run into some limits that you just can't overcome, even if you put a lot of effort into it. But if all those check out, you then move to the first step here, which is conceptual evaluation, where you'll use some of these conceptual tools to evaluate the sources of technical variability in the assay. And the biological relevance and all that stuff is very important too, but that's not what I'll be getting into in this presentation. And once you've done that, and I'll provide examples of many of these in the next few slides. After that's done, you moved on to within laboratory evaluation, such as control charting, looking at how your data actually looks across time. A lot of presentations I see, people show one replicate and things are looking great. That's very good in some ways of showing a proof of concept, but really the key is to show the results across time and how consistent they are. There's also robustness testing, histograms, and scatter plots. I'll show some of these in the next slides. Statistical data analysis based on the source of uncertainty encountered during the conceptual evaluation and the within laboratory evaluation. It's helpful to put together a statistical model to make decisions about whether something is a positive or negative, in addition to the probability of a false positive or false negative result. This is can't really be done as well if you have just for one number, if it's above or below it, it's a positive or negative. And lastly, you may need to show transferability. In some contexts of use, you may not need that. So there's advantages of doing interlaboratory tests, but in some cases that may not be needed. And I'll also highlight on the right side of here, there's a bunch of dotted lines. That's the purpose of that, is that it's not this linear process in reality. I've never had, theoretically, you could go through, do all the steps one at a time, and at the end you're done. But usually in my experience, you have to circle back, and it could be that you're doing the interlab study and you have to change some of your conceptual source of variability, or do more robustness testing, or change your statistical analysis. So these are all actually interrelated in practice. So here's an example of a cause and effect analysis. where the details aren't as important here, is more as a general idea. This is from an MTS assay, where each of these, in a cause and effect analysis, you kind of branch out what you think are the key source of variability. Each main branch is one of the key things. In this case, how you're handling the cells, the pipetting, the instrument, in this case, a plate reader, and so forth. And then through robustness testing and further evaluation, you could, or through control measurements, you could be evaluating some of these sources of variability. I'll go, since to my understanding, inhalation is a key part of the projects that we're talking about today. I'll provide a little bit of a case study or a use case where we went into a little bit more detail with this for inhalation-based NAMs. Or I guess we covered most of them. So one of the interesting things about the cause and effect analysis is that it can be a bit like Legos, where you have for different assays, there may be some shared source of variability. So for example, you may have a plate reader here among different assays. So if you have the source of variability well understood for plate readers, and you've done it with one assay, bam, the next time you use a plate reader-based assay, you're already a few steps ahead. The same thing is true with the exposure systems, whether that's pipetting, flow-through systems, or box systems like a cloud chamber. And also for biological test systems, this could be just a monoculture, a more complex system where you're making it yourself, or constructs, which are 3D versions. We do have some discussion about positive controls. I had a paper on I know that we talked a little bit about this during the last presentation and question and answer. Myself and some of my colleagues put together a paper on this that I'm happy, that if you're interested in getting to all the nuances for, or I think many of the nuances for selecting a positive control, we spent a whole paper just talking about that. So with that, then this was again an inter-organization collaboration. I actually did this when I was in Germany visiting the BFR for a month as a visiting scientist. So we kind of got talking about this, and it really made sense to work together and try to put this bit into practice. So as one example of what some of the branches could look like, this is what you could have for the exposure systems, where for closed box systems, these are some of the key what we thought were some of the key source of variability. Flow through systems, it's different, as is it different during pipetting. And again, once you have these the control measurements and robustness testing kind of worked out for these, you can really go faster the next time that you're doing it. We also in this paper describe common control measurements and some troubleshooting that could be needed. And all this was designed to help support the improving the technical quality and hopefully validation of some of these inhalation related NAMs. So moving on to another part of another conceptual tool, this is an example of a flow chart. What's not important here is the particular steps. What is important is that as when you think about these flow charts, the key thing is to have it so that your control measurements that you're taking during the course of the assay can cover the different steps. What you don't want to have is a black box where, say, step 3, we just don't know how well we're adding the chemicals or something like that, or we don't know how well our pipetting is working. Because in that case, if something's off in that step, you may not know it, and your answer could be off, and you would be unaware. This all fits then into the plate design. So in this particular, again, the details aren't that important. In this particular experiment, we were pipetting. So you'll notice that we have across within column 2, we had, within our 8-channel pipette, we have one single pipetting step. And then we also have a negative control in row A, column 2 through 9. And what was really interesting in this assay is that we end up finding for one of our probe molecules that it was actually photoactive, which wasn't in any of the previous papers. And the only reason we recognize that is because as we were pipetting it, we were monitoring what was happening in column A, and for some reason, it was far less in column 2 compared to in column 9. So this is just one example of some of the value of having a plate design where you're carefully specifying some of these things. What's nice is if you have a good plate design that kind of encodes quality into the assay. So if a new person takes it up, they don't have to necessarily think about some of these nuances in control measurements, but hopefully it's all really incorporated into the test itself. So here's what control charting could look like. I was mentioning this a little bit earlier, where, for me as someone who's interested in the technical quality, it's really helpful to see how the assay performs across time, not just in a single experiment. And here's examples of what we don't want to see. The first one is in part A is if it's declining across time, something is maybe degrading in your system or not working well. It's also not good as in part B if the uncertainty is getting larger across time. And in part C, there's a step change, which maybe some reagent was changed or something was happening there that caused abrupt change in the values. Scatter plots are really valuable too, and the idea here is that you want to compare among your control measurements. In this case, we're comparing against two control measurements. One is our positive control, which is cadmium sulfate in this example, and on the x-axis, on the y-axis is the mean optical density of our no treatment cell control, or basically, which is in this case was the MTS assay. So essentially, we're estimating our number of cells. And you can see in part A, this is what we want, where there's really no trend between the number of cells and the cadmium sulfate positive control values. What you don't want is in part B, where there's some clear trend there, where the fewer cells you have, the lower the cadmium sulfate EC50 is. So in this assay, we set our specification such that we stay out of the range shown in part B and stay only in the range for part A. So now I'll shift gears a little bit and talk about some work that I did with a number of colleagues from 8 different countries, and 20 different people were sending us excellent feedback for developing a guidance document for aquatic toxicity testing of engineered nanomaterials. This was done through the OECD. We had a bunch of meetings and a bunch of revisions, and after a lot of, I think we started about 2014, but we persevered, and in 2020, we were able to publish guidance document 2017, which is available for publicly available free of charge. I also published a paper myself and some of my co-authors, in addition Frank Vanderkammer, who had worked on some similar efforts for environmental fate testing, such as the dispersion stability. So that was published in Nature Nanotechnology 2 years ago. And so now we get into some of the more the nuances of what these covers, what it didn't cover, and so forth. Provide a little bit of an overview quickly for this document. Where first off, we're looking at the scope and why do we even need a guidance document? Why are engineered nanomaterials so different that the regular test methods may require some modifications? We described that a little bit, and then got into some other details that I'll describe in the next slides. So we have the one of the things so these are some things that we covered in the guidance document. One of them is the characterization of how to characterize the material, both as it's received and also the stock and test dispersions. One of the challenging aspects was how to monitor exposure during these experiments. There was a lot of effort put into how to make test dispersions. So how could you try to, what are some of the approaches that you could do to disperse the material and considerations for those different approaches? And if you did need to modify the test media, we give a hierarchy of changes you could make, as I described here, where particles are not sufficiently stable. And this was all done because a lot of the test guidelines, they specify that you need to be within 20% of the initial concentration. This is for those of you of us who've worked with nanomaterials, can be very challenging. A lot of them have the potential to agglomerate and sediment out of suspension. So this was a very tricky thing. But sometimes even if you put in all the effort, you can't reach it. So then you have to do time-rated averages and some other approaches. We also discussed control measurements and to avoid artifacts, and if needed, to understand the mechanism of toxicity. Sometimes you don't need to understand the mechanism for regulatory testing, but if you do, we provide some of those suggestions. There was assay-specific guidance for a range of the methods where we provide just general guidance and then kind of hone in on a specific method or two, or a method by itself, and describe some of the challenges there. And as I was saying in the previous slide, there's some things about dose metrics and whether we suggested, what to do. We suggested using mass-based concentration since that's the most widely developed for standards. Hopefully in the future, we'll have methods related to nanoparticle number concentration, but we're not there yet. We also discuss sediment testing, just and some of the nuances related to that. So just to summarize briefly, these quality tools will help enable more confidence in your measurement system. In this technical framework, this was really a group effort, and we hopefully provided some steps that people could follow that could help improve their technical quality if they want to take it to the next level, like it seems that based on the presentation I've been hearing so far today, that's definitely of interest. The plate design is a key part of that, and hopefully this OECD guidance document can help with nanoecotoxicity measurements and increasing confidence in them. And I guess I'll say I'm myself, very happy to have some conversations if people are curious about this or want to apply some of what I've suggested to their assay. I'm an email away. Having a conversation is a pretty low effort, so I'm happy to talk, and discuss any of these topics in more detail if anyone would be interested in that. With that, I'm happy if there to answer if there's any questions, and look forward to the panel discussion.
Thank you ever so much for that, Elijah. We do indeed now have just 30 seconds if somebody has a very specific question for Elijah. Otherwise, we will start moving on. Jacek.
Very quickly, Elijah, this is super exciting. I work more in the high-throughput screening of small molecules, and there is a lot of discussions on that actually aspect also in that world. So I wondered if you guys look into the literature and did you compare notes, if you will, with the other community, which does a lot of this kind of discussions also on plate, arrangements and concentrations and all of these aspects that we talked about.
Well, thank you for that question. Yes, we did. We've certainly been in touch with some of the our collaborators in the U.S. Who've been doing who had like a guidance document for how to produce these high throughput tests. Some of the nuances are a little, well, I'd say a lot of the conceptual tools would be overlapping. Some of the nuances are a bit different in that you have more wells to work with and you have it automated. So I think that there's some nuances there that differ a little bit versus if you're manually pipetting, but there's also, I'd say there's more in common than there is different, really.
Great, thank you. Very exciting.
Yeah,
thank
you.
Okay, one very last question, which is from the chat, and I'll ask the question quickly if we can answer it quickly. It's from Elise, and it says, seems that a lot of the conceptual tools are not only applicable for NAMs, but she also says, I now need to find where the question's gone. I believe the question was along the lines of, have you looked at random publications to see if you can test the data using your methods? And I'm trying to find the question again now, but it's...
Yeah, it seemed to disappear. It went into the ether.
Yeah, sorry, I suppressed it because I made a mistake in the wording, but that was exactly the question. Thank you, Sean.
So what Elise said, yes, these are general tools that, at least in my experience, could be applied to a broad range of assays. I haven't run into, maybe there's some nuances for in certain areas like high throughput testing where different things may be beneficial or possible. But in general, these are general tools that could be used. And I guess one of the things that when we got started off on this is there's an interlab test with the MTS assay that went south. There was quite a bit of variability. Maybe it was from the nanomaterials, maybe it was from the protocol. It wasn't really clear what happened. And then we took some of our colleagues in EMPA and had a second interlab and then started applying some of these tools. So I'd say that to some degree, yes, we've done this. And they help, I guess, is what I'd say is the takeaway.
Okay, perfect. Thanks ever so much, Elijah. I suggest you stay kind of on with your video on, and I'm going to hand over now to Blanca, who will introduce herself and the rest of the panelists, and we'll have just round about just over an hour, Blanca, before we need to wrap up. But I'll hand it over to you now. Thank you.
Okay, thank you very much, Sean. Can I share my screen first?
Yes, indeed.
I do have a
Have I already stopped?
presentation.
Okay, okay, good. I've been stopped.
Oh, great. So I don't see. Oh, here. Okay, okay, I'm there now. So I won't see you now anymore, but do you see now my screen in presentation mode?
We do, Blanca.
Great. Okay. Okay. So thank you very much. It is my pleasure to start now this roundtable discussion for our first day. Our session is entitled Harmonization and Standardization of In Vitro Models and How to Bridge Them into Regulation. And for this, we have our panelists here, which are our speakers. So they have been already introduced before. We have Valerie Swang from ECPAM, Tommaso Cerchi from NIST, Jorid Sørli from NRCWE, Alberto Katsumiti from GAIKER, and Elia Petersen from NIST. It's a really great group for discussion. My name is Blanca Suarez. I'm the Director of Regulatory Affairs at the Nanotechnology Industry Association. So I decided to put everything on a presentation mode because we are online, so it's fairly easy for people to realize and remember the questions. I will start asking the questions to the panelists, but then we also open the floor to have discussion with the audience as well, and to see if the audience have other issues they would like to bring on the table to discuss, if we have the time. So we have divided this roundtable in 3 themes. The first theme is the NAMs in the laboratory context. The second is NAMs in the industrial context, and the third will be NAMs in the regulatory context. So I hope we will have a really lively and interesting discussion. And with this, I will start with theme 1. And the first question that you can see on the screen, this question will go to, actually, since we just heard Elia, it will go to Elia and Valerie. And the question here is that NAMs represent a clear alternative to animal experimentation, at least partially. I don't think we are not in place now to completely avoid in vivo. Yet they need to be validated, and the path to validation is costly and lengthy. Is it possible to introduce shortcuts, perhaps some performance standards to streamline the validation path? Or do you have any other ideas to validation? I'm asking also Elia if he has some tricks or ways that he has a lot of experience in this. Some example here will be to reduce the minimum list of reference materials when we are validating, but do you have any other tricks for the audience here? We start with Elia and then Valerie, please.
Yeah, this was pretty much the whole intent of the framework I put together for improving the technical confidence, where I guess if you need to do an interlaboratory test, the more time you spent on the within laboratory and conceptual analysis, the better the chances that your interlab test is going to go well. So sometimes in interlab tests, if you have to do 2 rounds of testing because the first one doesn't go well, that certainly increases the cost and the length. So I would say, the more effort you do in first is better. Another thing I would suggest is, like I was talking about with the cause and effect analysis, as these branches are kind of filled out for different assays. If someone has a new assay, they can just really borrow it and hit the ground running. So that's another thing that can really streamline it. There are other, outside of my wheelhouse, there's discussions about, well, how many labs you need for laboratory testing and some of these other things that are, someone else can cover. I know there's work at OECD to revise OECD guys talking about 34 to get some more flexibility. But I'm a bit agnostic on that. I think with the lab comparisons, there's certainly advantages that you don't get from just having it tested within one laboratory, but for some context of use, maybe you don't need it. So, I mean, that's I think weighing among trade-offs there. I don't have a strong set of, well, you should do this or that. I'll open up, let other people chime in too.
Yeah, indeed, this was very much in line with your presentation, actually. And I don't know, Valerie, do you have any other, I don't know, issues with this, or could you give us some tricks as well?
Yeah, so validation does not necessarily need to be lengthy. I mean, if a method is mature enough to enter validation, validation can be quite quick, actually. Yes, I just follow up on what Elijah said. We are currently, we have taken the lead at the OECD to update guidance document 34 on the validation and international acceptance of methods for hazard assessment. So there we will try to streamline a little bit everything, let's say. But we identified, we think that the principles of validation, which is that a method or an approach should be relevant and reliable for a particular purpose, is still valid. I mean, we still want to have reproducible methods. We still want to have relevant methods. But the process to get there can certainly be streamlined. We have already performance standards. The concept of performance standard is already well known. And there are validation studies based on performance standards, which are carried out already since a long time. However, we would also maybe like to evolve the concept of performance standards and make them applicable to methods maybe which are not so similar, which provide the same, let's say, which have the same biological relevance, but from a technical point of view might be not so similar. Because at the moment, these performance standards are really applied to methods which are almost identical, let's say, like in the field of skin irritation, the methods on reconstructed human epidermis models, etc. But we would like to check if the concept could also apply to methods which are not so similar, let's say. And we have that already at the OECD. In the OECD test guideline program, we have already test guidelines which are grouped together according to the key events that they are predicting, let's say. So there the biological relevance and the mechanistic information that the method is providing is very important, let's say. Yes, so the guidance document 34, the OECD guidance document 34 was published in 2005, and the technology has really evolved a lot. We are faced with a lot of different methods, having also computational models inside, etc., artificial intelligence, etc. So we need to adapt guidance document 34 to keep pace with all these developments.
Indeed, I'm really happy that you mentioned this OECD document 34 because, I'm happy it came on the table, and I'm really happy that it's been updated as well. So
yeah, well, sorry, we are leading this together with the US and with the Netherlands.
So is there a date on the release of these updates, or?
We have our first expert group meeting. Well, it's called project group meeting in that context because there are lots of WNT members involved in that project group. We have our first meeting on the 5th of December and then probably a face-to-face meeting which will be back-to-back to the WNT meeting in April.
Oh, that's really great information.
Yes, and the leads have had already 8 meetings since we put since the project was adopted on the OECD test guideline program work plan.
Excellent. I don't see them. If anybody has any raised hands or anything.
There's a comment in the chat, Blanca, rather than a raised hand. And it's, can you please describe what is a performance standard?
Yes, so please, Valerie.
Well, it's already given here on your slide. There are actually 3 aspects in performance standards, which are the essential test method components, the minimum list of reference chemicals that should be tested, and defined reliability and accuracy values that your test should have. So basically, these are the three main components that you have in performance standards. So it's a bit a streamlined process to validate similar methods. You do not need to have such a large list of chemicals being tested. Usually it's a subgroup of the chemicals that have been tested in the reference method. And then you have this type of target values for performance. So within and between laboratory reproducibility and predictive capacity that your similar test method should reach to be considered as scientific valid. So we are working on this concept because these target values might not always be the appropriate way to go. Also, the minimum list of reference materials, looking more also at the chemical space that needs to be covered, etc. So we are working a little bit to evolve these performance standards as they are, but that's working process. So I cannot really give a lot of details about that at this stage.
Excellent. Thank you so much. I wanted to bring these topics on the table so everyone is aware and on the same page on this, and how we may be able to streamline a little bit this validation path. So thank you so much for both your replies. I think it's time to move on the next question, which I will ask Tommaso. Regarding reproducibility, should we invest more time in developing in vitro in vivo extrapolation models? Do you think we may have a lack of expertise here, or maybe we need to reach out to other disciplines to speed the development of models?
Look, it's complicated.
I know.
Life is always complicated. No, I mean, yes, we definitely need more in vitro to in vivo extrapolation models, possibly quantitative, if not only quantitative, because otherwise probably of limited use, even though any model that brings new knowledge is always useful. But I would like to make a distinction between in vivo. For me, that is absolutely true when we talk about human data. So you have epidemiology data, and then you have whatever test systems you have, you need to be able to relate it to our target. And our target, at least mine, I'm a human toxicologist, is humans. I'm a bit more skeptical if you need to develop new models, new extrapolation models towards in vivo animals. And also in that case, it depends. We know that there are some in vivo models which are working nicely, predictive, and that should be used. There are others that we know they are used because there is no alternative, but it is almost like non-using them. So in that case, I don't see why we should invest in extrapolating something to something that we know it's not so reliable. I come from a field of respiratory sensitization. There is no in vivo model which is actually working. There is the respiratory LLNA, which gives some information, but it is not designed for that. It is adapted, and we know it's not so predictive. So I would not use my time to extrapolate data towards the LLNA, in vivo LLNA. But you can extrapolate towards epidemiological data. So the answer is yes, but with some caution on how towards what you want to extrapolate and which data you use. Reaching out to other disciplines, it is not only advisable, but it is necessary. It is more and more interdisciplinary. When we do our in vitro inhalation models, yes, we have the cells, but then we have the physics of creating an aerosol, how you distribute. There is all the technology about the plastic, about the support that you use if a chemical interacts with the insert, if it goes through a membrane. Now we go into micro and nanofluidics, which is a different field. So yes, we need to reach out to develop the key vibe models, and also for that you need specialists in epidemiology, in AI, in all the disciplines that you need for modeling. But also when you develop the models, you have to be more and more interdisciplinary.
Yeah, indeed. Thank you very much, Tommaso, also for your views on the animal experimentation and these epidemiological studies. I don't know,
Sorry,
Sean,
Blanca, I just wanted
yes.
to make
Yeah.
a comment on the previous question.
Oh, yeah, sure, of course.
We, and I also agree partially that NAMS is in vitro, but the current status is that NAMS is it's much wider than that. So it's, because there are two acronyms, there is new approach methodologies and non-animal models. So, and they are kind of fused together, which is not entirely correct. So as NAMS, the most accepted definition to the best of my knowledge is that it is anything that it is new, preferably in vitro, but doesn't mean that if you have a new in vivo model which is more informative and so on, it's not a NAM. So from my point of view, NAM is not only in vitro; it is larger. We had a meeting in the NAMS for Nano project last week, and some of the advisors agree with us that we should extend NAM definition even to FISCAM methods. So these are also new approach methodologies for different aspects of the hazard and risk assessment.
That's a little bit, that's interesting because the definition I found for NAMS was always alternative to in vivo, so includes FISCAM, but interestingly, they don't have to be new methods. So it's a little bit different to what you're saying. So that tells me that the NAM issue is still under discussion, right? I see it coming from different ways, and the connotation is slightly different.
Yeah, maybe I can say something with regard to this. Yes, it was the first time new approach methodologies, that's what it means, NAMS, was given in an ECHA workshop. And it had a bit of a broader definition, as Tommaso has said, because it would also include new technologies that you can also employ on in vivo tests, like omics, for example. Well, this created a lot of confusion. And still today, there are people understanding under NAMS really only in vitro, in silico methods, and others who use the broader definition. So that's why we decided to call our status reports, for example, non-animal methods. So it's clear that it's not in the broader context, that it's really non-animal methods, so in vitro, in silico methods and models.
Okay, I will rescue the definition that I found and will share it with you. So Sanjay
You
can
find a
enlighten
lot
us.
of different definitions.
It's for sure correct. What you found is absolutely correct. It's that every person working in the field has his own opinion. So and it is not harmonized.
I think it was, I don't know if it was FDA or OECD or something. It was like a main definition, but I have to rescue it. I don't have it on top of my head now. So, but thank you so much for these comments. I don't know, Sean, is anybody else in the audience who wants to contribute to this
There's
question?
nothing in the chat and no hand raised, but if people do have questions, then they can either ask them in the chat or lift their hand, raise their hand up.
Okay, thank you. I will move now to the next question. We'll go to Alberto and Jorid. So the EU has invested large amounts of resources into development of NAMS for years now, and then we see it's been discussed several times, very few make it into the OECD or EFAN. However, and due to the ban of in vivo testing in cosmetics in Europe in 2013, we have witnessed the development and acceptance of NAMS addressing skin-related endpoints in a record time, really record time. So this example shows that with the right incentives, the road to harmonization may not take so long. So I was actually wondering, are we lacking a real market trigger in some of the NAMs we are researching now? I'm sorry for this, a little bit of a provocative question, Alberto and Jorid, if you could give us your views on this.
Who should we start? Maybe Jorid, do you want to start?
Sure, I can start. I would say, since I'm most familiar with the lungs, I think the skin probably was a lot easier. So we have the driver to create NAMs for the lungs and for inhalation toxicity, but we have such enormous complexity. So we are still discussing where to start, which part of the lungs do we want to look at, which one part of the lungs are we going to recreate, are we going to test every single part, are we looking for the effects that we see from inhalation on the lungs, or do we want to focus on what we have done so far with the animal methods to look at systemic effects? Yeah, there is a we need to target what are we going to model, and I don't think we have agreed on this yet.
Okay, so it's not the incentive issue, but the complexity. I mean, having said that, skin sensitization is quite complex and it was achieved, but I suppose, yeah, we have even more complexity here. Alberto, do you have any views on this, or anybody else from the panel?
Yeah, well, I would like to mention, for example, financial support as well, considering, for example, that this process normally, for example, take quite a while, right? Like to get to the point of a standard, as far as I know, and I think Valerie can correct me, but normally, for example, I think this in ECVN, natural, I think most of the financial support comes from the national content point, for example. So ECVN doesn't provide any financial support like to support the tests. And also, I think, for example, the OSD matters well to develop a guideline. So normally it's a process that takes quite a while, more or less like 10 years. So basically, in European projects, we don't have time enough, right, to develop the full standard. And sometimes it's quite complicated to get the connection between this first project and the second project, or to extend the financial support to take this test and go to the OECD level, for example, or to a guideline. So, for example, in our case, in the iCare project, we are in a very initial phase of the development of this standard. So right now we want to have a very solid base, for the methods that we're developing now. But we're pretty sure that, for example, during the time of the project, we won't be able to finalize. I mean, luckily, we will be able, for example, maybe to submit this to ECVN at the end of the project. But of course, this will probably take time to be, tested, for example, by the partners of NetWell, for example, and so on. So how we can maintain, like this financial support along this time, not just for the development, but also to the validation process, and then to the very end of the process, submitting, for example, to the OECD or ISO for a guideline.
Yeah, probably we may need also some private money, and that could speed things up. Perhaps if we get the interest of any of those companies who could commercialize the final test, maybe things will go faster. Perhaps I don't know if any other panelist wants to comment on this. I don't see any.
Maybe I can say something. I think everything that has been said is certainly true. I mean, it's always good to have the resources, the financial and human resources, of course, then things go much quicker. However, there is also this complexity question, indeed, that at the moment, we have a lot of mechanistic methods which do not provide on their own the full response, let's say, and we need to put them together in testing strategies in what we call, in OECD terms, defined approaches, or IATA's integrated approaches to testing and assessment. And we have for defined approaches, we have for the endpoint of skin sensitization, some defined approaches that were included in test guidelines and now fall under mutual acceptance of data. For IATA, it's a different question. That's more difficult to have it included in a test guideline, because there is a weight of evidence evaluation that is difficult to standardize. And also other aspects are difficult to standardize in an IATA. So certainly there is a question of resources that needs to be tackled, because as was said by Alberto, a lot of funds go into research and development, but less so on validation and standardization. So we have a lot of discussions on that topic in the European Commission. But also the complexity of the questions that for the systemic endpoints, let's say, we need a whole range of methods, of mechanistic methods that need to be combined together to give a meaningful answer, let's say. So yeah, there are different challenges. And of course, yes, if there is a change in legislation, like for example, the animal testing ban and the marketing ban of cosmetics tested on animals, then of course, this is triggering a lot of research and development and validation. So that's always very helpful, but it's not so easy to change legislation.
They are really very drastic, I will say.
Yes.
I don't know if there are any other comments.
There is a question.
Oh, okay.
Blanca. From the chat, it says, do you believe that NAMs will help us to implement the read-across approaches for nanomaterials? What are the challenges?
Anybody wants to get this one?
I can try to start. So yes, I do believe that NAMs can help in this. So the more data we have, the more easier it will be to make read-across approaches. For me, the biggest challenge is not even the method itself; is how to test the methods, and especially that is true with nanos. So what I mean is, while in the field of chemicals, we have a plethora of different chemicals with known activities, which can be very specific. So you want to test the binding of something to a receptor, you might find a positive control which you can use to benchmark your method. So that is not true for nano, not always. We really lack nano materials and nano references with known biological activities, which is very specific. And I'm not even sure if that is possible to do, but having to develop new methods, having unknown controls is very, very difficult. And what we did in the past was we started to test our methods with chemicals, and then you go to nano. But then again, if you're not fully confident on the robustness of your method, you test something unknown, you have to believe that the method is working, because otherwise, if you start saying, is it because nano is not compatible or because the method is not well developed, you enter in a never-ending loop which you cannot break. And we need to break this loop, and we need to ask the resources and the technology to develop, call it reference materials control. The semantic is not important, but we need benchmark materials and chemicals to be able to prove that our methods are universal. And the method should be universal, because if a method is designed to predict a certain outcome, in principle, if it is realistic and reflecting the physiology, it shouldn't matter if it is a nano or not a nano. And we are still not there. So once we reach this point, I think all the rest will be, at least in the field of nano, downhill, because then we will be able to use much more reliably our data, which we can use to do the read across, to come back to the original question.
Indeed, I fully agree with you, Tomás. So we are dealing with uncertainty to the power of 2. We don't know we don't know the results of the test, and we don't even know if the test is robust enough. So it's really hard to come up with a solution without these benchmarks or controls. Fully agree.
We have a couple of raised hands, Blanca, if you're
please,
ready.
Yes, please, because I don't see. Yeah.
The first is Jacek.
Please, go ahead.
Hey, thanks. I think I wanted to maybe ask you about the comments on something which I think connects a little bit the two things that you've just mentioned. So one is the fact that, there is no this well-studied, well-known, clearly performing materials as a reference. And another one is this like trigger or the incentive for us to do this sort of work. And I think we've heard now from Jorid, I think, that European Commission is looking into that. But I think the big problem in the research-based environment is that researchers are incentivized for doing something original, different, not the same, right? If we are doing things the same, then we can't publish that well. Then we can't do this, can't do that. And of course, if we just want to, and we here talk about the importance of doing things the same, of repeating them, of evaluating. And of course, if we can get resources from the industry, which is more interested, of course, in having something robust, then this might be a stimulating thing, because I don't see how just by the public funding and just by the research-oriented funding, we can actually create that incentive of just doing this cross-board, work. I don't know what are your thoughts about this, but I think this is you've been hinting into that, maybe not that directly, but when you have been commenting on the lack of trigger or maybe need of a trigger.
Yeah, exactly. And this is what happened when the ban on in vivo in cosmetics came in, and a lot of research was done by cosmetic companies themselves, because they needed to continue with innovation, which stopped for years because they didn't dare to, to launch products in the market if we didn't have the proper tests. So they did invest, and there was a lot of private money into that. So yeah, we were hinting that with the question. And exactly, I don't know if anybody has any other comments on this. I also agree with public money. We won't be enough resources, and also like, researchers are looking for innovation, and harmonization doesn't seem to fit, with innovative brains. So yeah, completely agree.
Yeah, there would be a need to have a different type of projects being funded for standardization and validation that would not fit under the R&D calls, let's say. But I think it could be feasible, but it's true that a lot was funded by industry when this animal testing ban and marketing ban came into place. They funded a lot of research and development and even validation.
Thank you. So it will be great, Valerie, if we have some calls, European calls, which are not research, but lead more into this kind of topics. That will be really very, very appreciated, I guess, by the community. But we should engage industry, I think, on this, and identify the who may be interested in a similar way as happened in the cosmetic ban, in the in vivo ban for cosmetics. That could be also a solution. Maybe tobacco companies want to invest in what Tommaso was presenting earlier today. No,
they want. Is that they encounter difficulties in having replies. So. And not only them. I mean, look, I'm not a paladin of the tobacco industry. We have worked with them because what we offered us to do was in line with our ethical conduct. We didn't test products or whatsoever. But I know many colleagues who would not reply to them just because it's tobacco industry or chemical industry. There are some colleagues who do not engage with the industry because they want to be fully independent, which I admire and respect. But then we cannot complain that there are not resources. We work with many different industries doing many different things. I think if it is transparent and if there is a clear framework of what you do, for what purpose, and for what use, that should be welcome. And you would be surprised over how many interactions you can have and how fast you can push things with the support of industry. Of course, we have to remain independent. That's normally what the industry wants. We don't want to have puppets, and then finally the results are not usable. So at least this is my experience. So, but there is money in industry, and even now they do. Of course, as Valerie said and you said, if there is a regulatory push, then the situation is completely different. I wonder if that was possible for cosmetics, because cosmetics are not simpler, because they're not simpler, but because they are not supposed to have systemic effects, because the technology was already a bit more mature, because that also what pushed the regulatory change, because it was not that one day our legislator decided that, okay, there is no method, there is nothing, there is no technology, but in 10 years we want everything. So it was a bit more advanced the field already, and then it was taking a decision. I wouldn't do it now for inhalation talks, to be honest, to say that in 10 years it should be animal free, because that would be a too high risk.
No, yeah, indeed. No, we should not ban in vivo yet. I think also we had there was a lot of public pressure in Europe to stop animal experimentation with cosmetics. Also, that had, the power of the people here. Also, was very strong against this kind of experiments. But I don't know. Maybe I see the chat moving up. I don't see the questions. I wonder, Sean, can you give me a hand? Or...
Yeah, Marcus did have his hand up. He seems to have taken it down. I don't know if Marcus still has a question.
I hope I'm not drifting too far away from the main topic, but in between there was the discussion about the number of reference materials to have a test system be validated. And I'm just experiencing very much trouble testing dispersion stability with multi-wall carbon nanotubes. And Tommaso, you mentioned this reference material. What's the number?
401.
MN401, and
Yeah.
I think they are quite short. And the kind of tubes I'm busy with, they are very long, and they are entangled, and right away they are not dispersible. So if you just in the beginning you cannot disperse them without destroying them, because if you apply too much ultrasonication, then they will break, and then you are testing another thing. Then I don't know if it makes sense. And so I would like to have some exclusion criteria defined for some testing, just in general, not only for in vitro or physicochemical testing, but also for ecotoxicity testing, stating that if a test material exceeds length sizes like, or is not dispersible at all, or so, then the test may not be relevant. So maybe you can stop testing here because of higher or top-down considerations you may, based on physicochemical aspects or other thinking, this test may not be relevant here. But ECHA insists having those tests performed. Another example is it's not very appropriate here, because it's for ordinary chemical, and it's an in vivo testing, chronic fish toxicity testing with a highly readily biodegradable chemical. And we are having a flow-through test. The test substance is not volatile, but it's biodegraded so rapidly that even in the flow-through test with a maximum media exchange rate, we are nearly not able to keep the concentration stable. So it's unreasonable to sacrifice vertebrate animals for a test chemical, which will never in the environment, in any river, be for longer time periods be stable enough to cause any chronic toxicity. And therefore, if we just would like to focus on some cut-off criteria in any testing, which make a testing unreasonable to be performed, this would be of great value for resources and for animals. Thank you.
Yeah, I'll respond a little bit. From the, in the OECD guidance document, there was a lot of discussion about, if something is so unstable, how do you do pelagic testing, of organisms in the water column? And that's why we also provided some discussion about how to do sediment testing. So, based on, I guess I know in the US you're allowed to talk with the regulators. I think in the EU there's some different rules with that, but that could be part of the discussion. Certainly for some carbon nanotubes, natural organic matter could help them stay suspended. But the problems you're talking about are, for quick biodegradability, could also be an issue with chemicals. So I mean, I think these are very real challenges, and I appreciate the struggle that you put into trying to resolve them. And I think there's some types of modifications or tests that maybe that could be possible to assess it. I think another kind of related thing that came up a lot with ICD-10's document is there's competing objectives. One is environmental relevance, another is worst case scenario. So to have something be the most environmentally relevant, you might want to have natural organic matter always present, but that often would not be the worst case scenario. So that's why we typically suggest to not have that natural organic matter there, because you could get a more conservative estimate of the potential risks. So, in some of your frustrations, I also hear some of these things of like, well, is it even really reasonable? And I think to some degree, they, in my, from my recollection of discussions, we try not to have it reasonable, we try to have it be conservative. So if it's not toxic under these conservative conditions, we can have some extra confidence of in the real usage, it would be still safe.
Okay.
Okay, thank you.
Thank you. I think we may need to move on unless there is any burning issue on these topics. Sean, do you see any other issues on the chat?
Just a quick comment really from JSEC, really saying that dedicated calls for harmonization, so reproducing results, seem to be a good important trigger. And harmonization, he thinks harmonization still is in fact is R&D work, although it's often not seen as that. And that maybe by informing sort of harmonization efforts, they could fit into the existing R&D Horizon Europe and national funding. So in other words, by changing by assuming that harmonization is R&D, it should be fundable, and you could have calls on that, which would then give you the support that's needed, I think, is the summary.
Okay, thank you. I think we should move on now to the next question, which deals with the non-validated NAMs, which are still, in my opinion, could be useful. If we are thinking about safe and sustainable by design approaches, maybe we can use them in the beginning of, early ideas. And I want to relate back to Tommaso's presentation today, and also Alberto's. Tommaso presented different types of equipment to kind of do the same to address the same endpoint, and each of them had different issues. And if I'm an innovator, I want to just test my material using non-validated some quick tests, but I'm not sure if that was quick what Tommaso presented. But how can we help industry to choose in this case you had 4 or 5 different possibilities to choose the best approach? Should we have an inventory of ongoing, I call them NAMs here, and the different status they are in, so we can help in industry or in academia to choose where to go, because we have so many of these different approaches, and people may get confused. Maybe Tommaso, you could start with this, and maybe Alberto follows.
You have a pleasure. Let me precise one thing.
Yeah.
I don't think you can choose between the best exposure method. You probably need all of them, which makes things more complicated because not all of them serve the same purpose. So if you want to be flexible, you need to be able, in a sentence, to expose whatever to whatever. So you want to have whatever material, solution, dispersion, dry material, long fiber, and be able to deliver it in the most appropriate way to the test system. And that implies, of course, your personal taste. So in macrame we had long discussion about continuous flow, yes, continuous flow, no, and I agree with our colleagues from BABA. Probably the continuous flow is the most realistic one. I have a concern there, but if you need to serve high throughput testing, it doesn't fit because of the throughput that you have, unless you redesign everything and then you design new machine. I'm a great fan, more than me, my staff in the lab, of the tech and digital dispenser and of the cloud system, because they are very fast and very reproducible also, but they have their limitations. So we have all of them, all the instruments that they have seen that they have shown in the slide. We have them in the lab. Now, I cannot imagine that every lab who wants to do it and does development can reproduce what we have, or that we can reproduce what other labs have, because we start to talk about consistent investments, which means that also the whole theory about having a round robin for validation with, I don't know, I say 10 labs, it becomes less and less possible, because you need to have millions of euros in investment only in the equipment to check if something is workable or not. So yeah, it is a very complicated thing to say about the inventor of NAMs. I want to quote, and I hope that it doesn't mind. I want to quote Maurice Willen. He said a few weeks ago, I saw one of his presentation, and he said, but we have to be careful, because it seems that the more NAMs we develop, the less NAMs we use, and it is true in a way. Exactly for what you asked me at the beginning. It is confusing. You go out there, you look for method for skin, and you find hundreds of companies, hundreds of different methods, which are all similar but not identical. And I think most of them are not well characterized in what they can do. So they're not so transparent that you know what it is your input and what it is your output and how confident it is. And we need, I think, in a way, we need to slow down the development and start really refining and understanding if what we have is good enough and how to use it. And we have some good experience with industry. We're interested in the methods, even if they're embryonic and they show something new to them, we're happy to use it and to test it and if possible, integrate it in their workflow. It's not a big issue for industry to use non-validated or non-regulatory ready method, as long as they provide an added value. And that's the thing. When you develop something, you need to make sure that the value is there and that you know how to show it and you know the limitation, because there are always limitations. And if you are transparent, then there is a use for it.
Yeah, exactly. That's why I was calling for an inventory, because I could put myself in the shoes of some consultancy people who may be listening today, or some research academia groups. And we have seen so many, only today, many different approaches. So yeah, they may think, yeah, but how do I choose one? Or maybe you're right, maybe you have to do all of them. Or yeah, and this is why I was thinking maybe we have an inventory where people could look. Or I don't know, Alberto, would you like to also comment on this?
Yeah, no. So I think it's very important to understand, for example, the limitations of the methods, as Tommaso mentioned, because, for example, some methods that we discussed like several times, that are quite complicated to adapt or to use, for example, for complicated materials, for example. So you can't basically use this method. So there is a limitation. So it's very, very important to understand the limitations. But I think I like the idea of, for example, combining assays to have like a better perspective. So the idea of using like IATAs, for example, I think is very, very, very important. We can't rely, the result in just a single assay, for example, in just a single method. I think it's important to combine different methods to provide like a more strong or a more robust, results. I think for industry as well. We discussed like several times, for example, on how usable is a method. For example, I think, for example, if a method is too complicated to be used, so it's another limitation as well. Well, in several projects we discussed about this. It's not just to have like a very good methods, but we need to try to be simple as well. We need to try to be, use simple methods or simplify the things. So this is a thing that we want to explore, for example, in the iCare project. It's not just like develop the method, but it's how we can simplify the method in a way that's not just scientists, but with very sophisticated labs can use, but also industry with limitations in terms of facility or equipment.
Thank you,
Blanca,
Alberto.
if I can make a small addition, and Valerie will forgive me for putting her in the spotlight, but there are some inventories.
Okay.
JRC made a scouting extensive review on the available methods for inhalation not so long ago. And I don't remember how many you found, 160, 180 models.
Yeah, this is mainly, that was mainly done for the biomedical research, because we are not only working in the regulatory field, let's say regulatory toxicity, but also for, in the field of biomedical research. And it was really to help the how do they call that, the national committees that are set up in the member states with regard to the directive 2010/63, because they do not necessarily know what is around as methods. So in order to help them to retrieve that information, we have set up these inventories in various human disease fields, let's say. And one of those was, I think, yes, respiratory. Yeah. There are other fields. So yeah, these are a type of inventory. There are other types of inventory. I mean, we have ZA, as I said. This is, for example, this database includes methods that have been submitted to us and which did not necessarily go through all the steps of validation and afterwards regulatory adoption. So there you can also find information. And then I think there are also databases from DGRTD, which have all the method description or the SOPs. I don't know how much detail is in there of methods that have been developed in EU-funded projects. So you have a lot of these different inventories, databases, and there are also currently a lot of discussions on this. Would there be a need to have a centralized database where all the information can be found, etc.? So this is all these issues are actually under discussion, I must say, and there is no one size fits all solution that has been be found yet. So
okay, but we have an answer to our question. So there are some inventories, which is really interesting to hear. And I don't know, Sean, are there any other questions in the chat?
There's one question, which is, are there cost aspects to be taken into account when the selection stage comes?
Selection for whom? You mean Declan or?
I presume more selection of which NAMs someone would choose to use.
I will say, I mean, from an industrial point of view, yes. I mean, we ask for different quotations, and yeah, in my opinion, for industry, yes. But of course, the quality of the results should be also taken into account. Because to be honest, I mean, I can actually comment a little bit on this. We did an exercise some years ago comparing the cost of in vivo skin sensitization against in vitro, the in vitro array of assays. In vivo is cheaper than the three of the in vitro together. However, there is a since threefold increase now in the in vitro skin sensitization from less foreign companies in enriched dossiers. So it means here that even if the in vitro is more expensive, people are using it more often now, because I think the use went threefold up. So yeah, this is just a little insight coming from my side, from what we have seen. So yeah, maybe not always the maybe people do take into account also ethical issues as well. But I mean, I open the floor for these two others to comment as well.
But from my point of view, the program cost is very relevant. We have a tendency to compare NAMs one-to-one to in vivo, which is true for maybe one endpoint. But you cannot imagine that for every biological endpoint, you have to run a full NAM, because otherwise the cost will simply skyscrape. Yeah, I think we have to find framework approaches and IATAs where we start from in silico prediction, which direct the selection of the NAMs, to keep the cost reasonable. Otherwise, it's not going to be feasible.
Thank you, Tommaso. And Sean, any other issues on the chat, or we can move on?
Yeah, there's just a comment on infantaries from Eric Bleeker, which is infantaries are nice, but then guidance on when to use which NAM appears essential.
Yeah, sure, of course. Otherwise, yeah, we hear about 160 assays on the inventory that were mentioned before. And I wonder if that helps anybody to have 160 without any guidance. Indeed, I agree. Okay, let's move on. Yeah, I mean, this was on the based on what we have seen today. We see how complicated NAMs may be turning into. We need to, I mean, some of them high content analysis, artificial intelligence, and when one wants to speak to regulators about this, they probably are not experts on all this. And also thinking about traditional toxicology, maybe they relate to dose response kind of points rather than having this kind of outputs, which are complex. So our regulator, Andrea, unfortunately is sick, so she cannot join us today. But my comment here was that we should be heading in the future for also the regulators of the future, a group of multidisciplinary people who have come from different backgrounds. So it's easier for them to understand the NAM and accept it. I was at ECA yesterday, and I had this discussion with one lady who is a regulator, and she of course fully agreed that they now, many of them, they don't have the knowledge sometimes to understand these complicated NAMs, and we may be losing time and also in interesting ways of doing the new research on toxicology. But anybody wants to comment on this?
I think the level of understanding of regulators in NAMs is a bit different. It depends how much they are also engaged in evaluating those, how much they're engaged in the validation exercises at the OECD level, etc., etc. So there are regulators who have a good knowledge about NAMs because they are engaged and they keep pace with the progress, and they try to stay involved, etc. So but certainly like everybody else, they certainly would also need training. I think I also need to keep myself up to date all the time. So yes, it's important that everybody gets appropriate training and stays informed, especially if you have to take decisions on the basis of this new technology. We should also, maybe in your first paragraph here, we should also now think a little bit differently and not necessarily want to predict the adverse outcome in an animal by using NAMs and combining these NAMs. Maybe we have to have a result that is allowing to protect consumers, environment, etc., rather than predicting the adverse outcome of a current animal toxicity test. That's also, I think, a shift of mentality that needs to happen. We do not necessarily need to have exactly the same results as we have in an animal test. As was said also before by Tommaso, we want for human health effects, we want to protect the humans. So we need to know what is happening, what the adverse outcome is in the human and not in the animal. So this needs to be a bit, the mentality also there needs to change. And we must say that regulators are used to see the animal data. Especially the older regulators, they always took decisions on the basis of animal data. Now they get other data submitted to them, and they have to interpret them and evaluate them a little bit differently, let's say. So But I think if everybody works together, I think we can get there.
Yeah, exactly, because we've been hearing about this now for years, but I don't know if we are really moving very slowly or things are happening in the background that we don't know, or yeah.
But there are programs like, for example, APCRA, which stands for Accelerated Pace for, I can't remember the acronym, risk assessment. They are the regulators of the different jurisdictions work together to really develop case studies where they develop IATAs, and they check how methods could be combined and what type of decisions they should take, etc. So there are some programs that are already ongoing where regulators try to work together to better understand how these methods can be combined and evaluated, and what type of data they generate, and how these data could be used in their regulatory contexts.
Okay, that's good to know.
Maybe there should be more of this, but some
good to know.
Yeah, some things are ongoing already.
Thank you. And I have another question, and this is I allow myself for this because we bump into this problem when talking to some companies, and I wanted to see if we could have a little discussion, if you have any comments. I mean, we don't have Andrea here in the panel, unfortunately, but there are some regulators also in the audience. I don't know if you know about the EFSA transparency regulation, which requires that all studies performed for a particular product are made available to EFSA. My question here is, do you think this kind of regulation prevents the validation of NAMs with potential market products and hence limits their widespread use? I'm asking this question because if you are validating a NAM, which is not fully validated yet, and you use your future product there, and your results will need to be made available to EFSA. But this NAM is still under development, so it doesn't really mean that your results are clear-cut. So this issue came up in different groups of discussion groups. And I don't know if we have here anyone who could comment on this. Otherwise, we will jump over this question. But I was just curious because we had to link also regulation and NAMs to see if anybody had any comments on this. Or this was completely not interesting for you, or you didn't know about this. I don't know if anybody wants to talk about this. Sean, do we have any comments or?
There's no comments on this specific, Blanca, but there is. If Valerie could just sort of clarify the program that she mentioned.
Oh, sure.
There's
yes.
a couple of questions on just clarifying the program's name.
Yeah, I will write it in the chat.
Thank you, Valerie.
I have to check. Maybe Elijah knows. Accelerating the pace of chemicals risk assessment. But these are the regulators. There are regulators from the EU. ECHA is involved in that. EFSA sometimes as well. And then the US EPA, Health Canada. I have to check who else. This is a voluntary initiative that was started among regulators.
It's just been put in the chat, APCRA.
Yes, APCRA, accelerating the pace for chemicals risk assessment.
Thank
Blanca,
you. So
just to give you some support on this, it is a personal vision. I'm not a regulator, so I cannot speak for them. Honestly, I believe that transparency is a plus, is a value. So everything that we do, especially when we talk about safety and risk assessment, should be as transparent as possible. And from my personal experience in the interaction with regulators, I think they are open to explanation, and it's not that because you have presented some preliminary data which are not so good, then your destiny is doomed forever. I think You can explain, you can exclude some data point if it is not reliable, and you know why it's not. So I don't think that this particular point will hinder NAMs or product development at all. I mean, but at the same time, it gives consumers, or it should give consumers, the trust that everything that is known is put on the table, and we think about the whole process, developments, consequences, rather than allowing to take a decision based on one study selected carefully among 100 studies. But then I believe that everything will be evaluated all together, and the way of evidence, even for the quality of the experiment. So if there is some problem in an experiment, for sure it will be discarded.
Yeah, thank you, Tommaso. And this is also being discussed at ECHA as well. So it may also appear there too, following the EFSA experience. And yeah, they are looking into that as well. So,
but sorry, I don't really understand why this would prevent the validation of NAMs.
It will prevent in the way that if you are developing a product and you want to use it, like for example, for a food contact, well, this is what I have heard, for a food contact material, and you use one of these NAMs, which are not validated, so you don't have any assurance of the results, how accurate your results will be. Yet, if you want them to file this thing in EFSA, you have to give also the results of that experiment, which you have performed with a NAM, which nobody gave you really 100% sure, and and the results, assurance that the results will be accurate. So you may be experimenting with a NAM because it's cheaper than going into the standard test, but maybe the results may not be good for you because the NAM maybe was not suitable purpose for your product, but yet you will have to send this to EFSA. So we were wondering if industry, in this case, will steer away from NAMs because they may be scared they have to submit everything to EFSA. And this was where the problem came from. So I just wanted to hear your opinion, but I also hear Tommaso. Of course, transparency is always good. But we may be looking into a problem like this or may not. I just wanted to have this here for your consideration. Yeah. I don't know if it's clear now what we meant with this question. So I think we don't have a lot of time left. I do have some other questions here. Oh yeah, I think this was already discussed previously. If in vivo should still be the gold standard. I think it was answered also by Tommaso earlier. When you talk about the epidemiological studies, and anybody else wants to comment on this?
Yeah, I also already commented on this before that we do not necessarily need to predict the adverse outcome in an animal with our NAMs, that they should help to protect consumers' environment, but that doesn't necessarily mean that they need absolutely to predict the adverse animal outcome that is currently required in regulation, in the REACH regulation, for example. So I think we have to move away from this because the NAMs are giving slightly different information than the animal data, but it's not therefore that it's not good data.
Yeah, indeed. I don't know, Sean, do we have any comments on the chat on this?
There was just one comment from Eric again, where he was basically commenting that for the body that was mentioned earlier, APCRA, why only regulators? For me, this raises the question on how big the gap is between method developers and regulators who should interpret the outcomes. Method developers should be able to explain to regulators how their method can be used, and regulators should be able to explain the questions they want answered.
Yeah, maybe that takes place before they come together and they discuss on how to combine these methods to be useful in their specific regulatory context. So I think these type of interactions are are happening, but not in that APCA framework. Maybe this take place before in other frameworks.
Okay, so yeah, there may be other places where this happens.
Yes.
Okay. Mm-hmm. And yeah, my final question was on the nano-size controls, which we have discussed for a long time and keep coming up in different meetings. And I think also was mentioned today, ideally we should have nano-size controls, but it's not so easy. Sometimes probably they are experiment dependent as well. I don't know if anybody wants to comment on this. Please feel free to step in.
Yeah, I guess it depends on what you're trying to, I mean, the purpose of the controls. If there's something about the assay, such as for example, if you're thinking the part, I mean, because I'm trying to think of, would you need a control? I don't, off the top of my head, think of a time when you need to have, it's a positive control, something that's nano-sized. I mean, the positive control one of its main functions is to assess that the biological response is similar to what you've had in the past. There are other things that nanoparticles can do, such as absorbing chemicals, but that could influence your result, but you could just do that with your nanomaterial that you're testing. You wouldn't necessarily need a control to test that. So I don't know. I mean, I could be convinced that there was a case you need a nano-sized control, but I can't think of any off the top of my head where I'm like, oh, this assay would be a ton better if I had a nano-sized control instead of something else.
Yeah, the Ames test. I think it was the one and only case that we keep coming back to.
Oh, that's great. Yeah.
This is probably why this issue is still ongoing. But yeah, actually, to my knowledge, yeah, it was the Ames test, right? That triggered this discussion several years ago, right? So, and we still have this on the table. So, but
Yeah, but the Ames is very, very specific. So
yeah,
that
exactly.
works. And also, I mean, again, we want to predict human relevant endpoint using organisms which are extremely far away from humans. So I would be a bit careful there. I do agree with Elijah. I don't think we necessarily need nano-size controls, but if we do need them, they have to be specific for biological effects that we want to see. Otherwise, they're irrelevant. But as I said before, we need to gain the trust in our systems. So if a system is robust, then from my point of view, and again, I agree with you, we don't need necessarily nano-size control. We need them because we're asked to get them. So you go and you're asked to test the nanomaterials, and they ask you, have you used the nano control? And then it's better if you have, because then otherwise you have to answer no, and then you have to go back again. So it's more a matter of mentality and lack of trust in the methods, from my point of view.
Yeah, thank you, Tommaso. I mean, Eric, would you like to step in as well? I heard there was a comment on the chat from Eric.
Yeah,
I think I can try. It's on the topic of do we need nano-size controls? It really depends, I think, on the method that you are looking at and what that should work with. The aims that you mentioned was one of the things that it definitely matters whether you're looking at a nanomaterial or something else, but for others it will probably be less relevant. So I totally agree with what was said before. But what I'm missing a bit, and what I put in the chat that Sean read out, is it still seems a lot like we have developers on the one hand and the regulators on the other hand, and they maybe talk to each other, but not necessarily that often. Also in that ACREP or whatever the acronym is, I can see that regulators want to talk among each other for certain things, but having it sounded still very separate, like, okay, we have talked to the method developers, and now we can talk among ourselves how useful this is without going back to the method development. Like, okay, nice that you developed this and this is useful for us because of this. This is not useful because we
cannot use it in this and this context for this and this reason. I think those type of discussions should be there, and it still, I feel a lot of different groups that don't talk to each other, and I don't think that brings us much further.
Thank you, Eric. Excellent point. I was thinking about these trusted environments that are being developed in different EU projects. Perhaps, yeah, that's a topic to take into those, right? To try to bring together more discussion with different stakeholder groups.
Yeah, can I maybe, from my experience, the test methods that are submitted to us, we very often notice that the test submitter doesn't really know how to use the method in a regulatory context, and they really need advice from regulators. Yeah. So as I have presented this morning, we have therefore set up this network of regulators, Parere, preliminary assessment of regulatory relevance, where we can engage with the regulators already very early on in the process. Because these test methods look really nice from scientific point of view and sometimes also very well developed, etc., but it's not clear how you use it in a regulatory context and for what purpose. So, and it's also difficult to ask the test method submitter or the test method developer to give us that information, because very often they don't know. So there needs indeed to be some interactions between regulators and test method developers, because they have different interests and they have different knowledge, let's say. And we try to remedy by having this type of interactions with our Parere network. We also ask Parere sometimes about the regulatory relevance of methods or approaches that are developed in EU-funded projects. Like this year, we had a joint workshop between the ASPIS cluster and Parere, where this type of discussions took place.
Thank you, Valerie. I think we don't have any more time for discussion. Unfortunately, it was really interesting. I learned a lot. I hope you did as well, all of you. And yeah, as always, we need more time. It's always a lot of interesting topics to discuss, and we never have enough time. But I really thank you so much for your very proactive contributions. I'll give the floor now to Sean, I believe.
Thank you ever so much, Blanca, for chairing that very interesting roundtable. Just a little few comments from me, just to sort of summarise. It's obviously, I was asked to summarise the day. We have run over time, so I'm not just going to replay everything. But I would just like to say I think it's been a very interesting day where we've moved from learning to see what's still ongoing at the OECD, having a look at what's coming through in some of the pre-standardisation and some of the work that's being done in groups such as ECVAM, for example, and then started to get into some more detail around some particular NAMs that are being developed in certain projects, and then a final discussion, which I think is nicely wrapped up the day, and as Blanca said, has probably left us all wishing that we could continue for some time. This workshop first began in the NanoHarmony project several years ago, and it's really nice that we're sort of continuing in that vein of having this online two-day workshop. For those that are still interested in learning a little bit more, and for those that are still sort of feel that they want to learn a little bit more about the sort of whole test guideline process, one of the outcomes from the NanoHarmony project was indeed this process mentor that's been mentioned several times. I've just put the link in the chat. Feel free to go there, have a look, share it with colleagues. It gives you a nice overview of the whole OECD process and allows you to sort of understand the various steps that the various methods have to go through to become into a guideline. And all that's left for me to do is to say, I hope we'll see all of you here again tomorrow. We will start again tomorrow morning at 10 o'clock Central European time for a second day of this workshop. It just is left for me now just to say thank you ever so much today to all of our speakers who have given really nice presentations, who have been very open in answering the questions. Thank you so much to especially to the people that took part in the round table for looking at a lot of issues that were being discussed. And finally, thanks of all to all of you who have actually managed to join us here today. And I look forward to seeing many of you here tomorrow when our colleague Ernesto will be taking over the chairing. Thank you ever so much, and good evening, good morning, good afternoon, depending where you are. Thank you.